Okay, Assalamualaikum and good morning to all, especially to my supervisor, Associate Professor Dr. Ina Salwani binti Mak Yasin, to my coordinator Dr. Zaira binti Muhammad Zufri, and to all my lecturers and also to all my fellow friends. Okay, today I am Nurul Atiqah Muhammad Razi 191728 will present about my final year project. The title is Characterization of phatogenic Aeromonas spp isolate from diseased red hybrid tilapia. So this is my outline. So Malaysia aquaculture production contributes a significant source of animal proteins for human consumption in the country. Data from FAO 2019 reported that from aquaculture sector has increased to supply the demand from the consumer in 2016 until 2017. Aquaculture strives to produce high quantities of fish reproduction economically. However, the infectious diseases are one of the major of limiting factors for production. Bacterial diseases are the main pathogens that are responsible for severe disease and outbreaks in culture fish industry. Motile Aeromonas septicemia is the one major limiting factors, that also called red spot disease or hemorrhage disease, that causes higher losses for farmers up to 80% mortality, and its pathogenicity is mediated by different extracellular protein. So this is my problem statement. Aeromonas infection is one of specific incident that many cases either go undetected or are not reported, and the data from molecular epidemiological is a measure for preventive measure will be taken in this study, and the mechanism of pathogenicity that unclear and complicated that are needing to be studied and understood to avoid the outbreak of disease. So here I have three objectives, which is 1. To characterize the genotyping of pathogenic Aeromonas spp by using GORB genes. 2. To assess the virulent genes of Aeromonas spp. 3. To analyze the antimicrobial effect of selective antibiotics of Aeromonas. So this is my hypothesis. So for literature review, the mortality caused by Aeromonas is depend on the concentration of bacteria, fish size, and the type of bacteria strains used, the onset of clinical signs, and the pathogenicity. Although motile Aeromonas have received much bad reputation as fish pathogen, the virulence and capability of Aeromonas is one another important pathogen in tilapia culture in Malaysia in the future. So this is for fish health and disease management. So for this study, I'm using molecular methods because molecular methods is more accurate for identification and can be rapidly, and it more specific than traditional methods. So here is the major Aeromonas species. I have 6 species. So disease caused by Aeromonas have several forms such as peracute, acute, and subacute. So mainly caused by Aeromonas hydrophila that can cause 80% mortality, and this pathogen can cause damage in host and cause disease to human as well as animal. So the one of the symptom such as hemorrhage, base of fin, lost appetite, and so on. So for virulence genes, virulence factor of Aeromonas, I'm using 6 of virulence factor. That this 6 factor is major virulent genes produced by Aeromonas. So this is my location of study, which is I have 4 location of study in Perlis, Perak, Selangor, and Johor. Okay, from this study, 10 isolate of Aeromonas was suspected from various tilapia farms, and isolation and bacteria culture, and then I make a DNA extraction by using genomic DNA purification kit, and the detection and quantification I'm using gel electrophoresis, 1% agarose gel in buffer, to confirm the concentration of DNA product. So this is my PCR amplification by using GORB primer. So for this GORB have 35 cycles, and for primer GORB for forward and reverse, the product size is 1100 BP. So this is the PCR amplification for detection of virulent genes. Also same like GORB, which is have 35 cycles, and this is the gene which product size. So DNA sequencing stands for sequencing NCBI, that was analyzed by using the BLAST program. So this is the antibiotic resistance. So for antibiotic, I'm using 6 types of antibiotic, which is ampicillin, gentamicin, clindamycin, nalidixic acid, rifampicin, and streptomycin. So for antibiotic test, the pure culture was grown in TSB, and the Aeromonas strain will test against the following antibiotic discs, and after enrichment in TSB for 6 until 8 hours at 37 degrees Celsius, the culture was treated on agar plates using the cotton swab, and after 30 minutes of predation time, the plates were incubated again, and the diameter of inhibition were taken. So this is the prevalence of Aeromonas isolate from tilapia for each farm. So this is for phenotypic identification. So I have 6 biochemical testing, which is gram staining, oxidase test, catalyst test, motility test, indole test, and sulfide test. So as we know, Aeromonas SPP is a gram negative that consists of pink color and root shape, and when the oxidase strips was turned to blue color, so all the Aeromonas SPP is positive. And the catalyst test, the Aeromonas SPP was turned to positive result that show the formation of gas in the foam bubbles. The motility test, indole test, the organism is motile evident by a diffusion zone of growth extending out from the line of inoculation and grow throughout the entire of medium, as you can see here. Indole was produced a pink coloring, and as you can see the pink coloring, and sulfide test turned to black hydrogen sulfide as a gas indicator. So this is the summary of phenotypic identification for 10 isolate. So this is molecular identification, which is from GORB primer. 4 species of Aeromonas were identified from all 10 samples, which is Aeromonas hydrophila, Aeromonas veronai, Aeromonas jandi, and Aeromonas decensis. For this GORB, the agarose gel electrophoresis of GORB have 1100 BP product size. So I'm using the DNA marker 1 kb from First Base Malaysia as a marker. So this is the GORB sequencing analysis for 10 isolates by using the NCBI. So you can see here the query cover 100%, and percentage of identity also 100%. So this is the summary of identification. The total percentage of Aeromonas, as you can see here, 30% from 100% is from Aeromonas hydrophila, Aeromonas veronai, Aeromonas decensis, and 10% from Aeromonas jandi. So this is the virulence genes assessment. As you can see here, 4 out of 10 sample were positive with virulent genes elastase, which produced the expected size 513 BP on the gel. The agarose gel for cytotoxic enterotoxin, you can see here 3 out of 10 samples were positive with these virulent genes, which expected band 500. For flagella, the agarose gel electrophoresis were positive, which 2 out of 10 samples, which is produced the expected band 608 BP on the gel. So for lipase, the result is all the 10 sample is negative with these virulent genes, and for hemolysin 2 out of 10 sample were positive with these virulent genes, which produced the expected band 597 BP on the gel. So this is the summary of virulent genes profiling. So as you can see here, a significant number of Aeromonas sp has been described, including ELA, flagella, lipase, and hemolysin, but Hydrophila, Veronii, and Dakensis is more significant in number. So this is antimicrobial susceptibility test category, which is have 3 classification of zone inhibition: resistant, intermediate, and susceptible. Which is resistant is isolates are not inhibited by the commonly achievable concentration with the normal doses. Intermediate, the same antibiotic may not be adequately effective against the same organism, and response rate may be lower than susceptible isolates. And susceptibility, a category that implies that isolates are inhibited when the prescribed dose is used for the site of infection. So this is the detection of antibiotic resistant phenotypic profiles. So as you can see here, for ampicillins, Aeromonas spp are uniformly resistant to ampicillins, which is the zone of inhibition. Diameter of zone inhibition is the same for all ampicillin. However, so this is the antibiotic susceptibility profile. So however, another antibiotic, Aeromonas was sensitive to nalidixic acid, kanamycin, gentamine, and rifampicin. So for discussion, molecular identification by using PCR method was sensitive housekeeping gene such as GORB. A study conducted by King Gombe et al. In multiple PCR method for detection of Aeromonas reported that Aeromonas produce a large number of virulence factor. By using BLAST program, it showed that GORB are highly specific and sensitive, as the sample was identified at the highest similarities of 100% identities to the data recorded in GenBank. The virulent genes are the main factors for the bacteria being classified as pathogen harmful to the aquatic organism, especially to fish, and important to determine the pathogenicity of the bacteria in the water. The antimicrobial test in the study can find out the ability of other microbes to resist the effects of antibiotic. So the conclusion, the GORB show high sensitivity and specificity for characterization of Aeromonas spp. The virulent genes test was important to determine which Aeromonas species that are pathogenic and cause severe damage in freshwater fish culture. The prevalence of Aeromonas in tilapia and the distribution of multi-drug resistant strains that could be concerns to the farmer in Malaysia. The data that was collected in this study hopefully can be further used for management plan, and that can be applied in agriculture environment. and medicine sector. So I think that's all from me. Thank you.